here and you're kind of thinking you're pretty sure you're going to drop class, you probably wouldn't be here. Get out. Yeah, get out sooner than later, then you can make, I'm not trying to run you out, but if you're juggling your schedule, you're either going to get out of this class to make room for some other one. Try to do it sooner than later, then we can get these other people that are on the waiting list. Remember, there's another one, Tuesday, Thursday, and I think there's one in the morning. Oh, it's in the afternoon? Okay, that's during the regular day time. All right, has anybody got any questions from last time that you want to clear up before we move on? Nothing, we're in good shape? All right, so who takes good notes? Where should we stop? Yeah, right now we're talking about HIV, the human immunodeficiency virus, and the problems it causes in people. And we're going to look at, one of the things we're going to look at is the importance of an evolutionary perspective in figuring out what the problem is, but then also how to solve it. But specifically, I think we were talking about pathogens, bacteria, viruses, and things, and other parasites jumping from what? Non-human animals into humans. Is that a rare thing? No. No, that's in fact almost all the big diseases that you can think of. That's how that happened. They're in other organisms, and as we'll see, they're always making new individuals, and they're making variants on themselves. And humans are in close association with these other organisms. There's lots of opportunity for them to encounter humans. Now, if some of those variants can deal with humans as a host, then they're successful. And over time, they get better and better at dealing with humans. And then now you have some new pathogen, some new parasite that can deal with humans as a host, and they become established there, and then they move around, and it can be a big problem. So that's a common thing, and that's how human beings got this HIV. Well, it's a virus, though. So we have other viruses, and some of them cause some fairly nasty diseases, but quite a few of those we can deal with them. How do we deal with them? Yes, one of them is vaccines. And so how many people have got your flu shot? Or how many people get flu shots? And this is the time of year when you're supposed to get them and stuff like that? Yeah, and so you're preparing yourself for the future. Okay? Well, one of the problems with coming up with medicines or means of combating viruses, one of the reasons they're so problematic and regular antibiotics don't work is because they're really using your metabolism and they're using your energy and your chemistry. So you really can't mess them up too much without doing what? Yeah, without causing harm to yourself. So if you're dealing with some pathogen that's a bacterium, that's a whole different story there, because they have different kinds of cells than we do. They have prokaryotic cells, we're made of eukaryotic cells. So there's some differences there that we can exploit. In dealing with viruses, that's really, it's a different situation, so we can't do that. And so one solution is to do these things, use vaccines. And we'll talk about some problems with vaccines and an example of, say, flu shots and things like that. But when people figured out that HIV was a virus, one possibility to deal with it is by a vaccine. How long have people known about this? It's actually a process called variolation. How long have people been doing that sort of thing, or known that you can kind of fight the problem with the problem? It's actually for a long time. It really wasn't until recently that people realized, remember we talked about theories and the microbial theory of disease, learned what viruses are and bacteria. But almost 1,000 years ago, people did things that actually combated viruses. So there were people in the Far East. There was a Buddhist nun almost 1,000 years ago. And one of the diseases they were dealing with was smallpox. And she would take scabs from old smallpox pustules and grind them up. And I think she would blow them and have people inhale them. And lots of the people that she did that to didn't get smallpox, okay? So there was some, of course, they didn't know what was going on, they just knew that it did that. And soon after that, in places like China and India and Turkey and the, hundreds of years later, this became a fairly well-established practice. And then it moved over farther into Western Europe, and people were doing this and very similar kinds of things to treat or to prevent smallpox, okay? But still, some people died even though you did that. So you did this to a bunch of people, and about 2 or 3% of them, they got the disease and died because of what you did to them. And so for those people, it wasn't so great, but it dropped the mortality rate from that disease something like 10-fold. So in the big picture, a lot fewer people died from smallpox. Now, if you were one of those few percent that didn't do so good when they did that to you, well, that's bad, I guess. But it was really getting at this issue and solving this problem. But again, people didn't really understand what was going on. Later in the 1700s, there was an English guy named Edward Jenner, and he noticed that women that milked cows that got a similar disease called cowpox, they didn't get smallpox. So there was something about them that made them immune to smallpox. So what he did was he took some material from one of the pustules from a cowpox pustule, and he injected it into a little kid. It was like an 8-year-old boy. And then he waited 6 weeks, and then he exposed this boy to smallpox, and he didn't get the disease. Okay? So that's what variolation is. And a lot of people, it's like you're taking this problem and you're injecting it to people. A lot of people were not very happy with that, disagreed, particularly that kid's mom. I bet when she found out, you did what? But it worked, and he was protected from that. And what's going on now that we understand, what you're doing is you're taking either a virus that's very similar, or what we do now is we take viruses that are very specific to certain diseases, and we render them inactive. And then you put them in somebody's body, and then your immune system, okay, builds up antibodies to those antigens, and it's like building up your army that's very specific to a certain enemy. Then in the future, should you get exposed to that particular enemy, your defenses are up, and you knock it down, okay? And so that's what vaccines are all about, okay? Where does that word come from? What organisms were the reservoir host for smallpox and cowpox? Yeah, what's Spanish for cow? Yeah. That's where this word comes from, okay? Vacinas is actually of a cow. So this thing that he was doing was coming from cows, and so that's where we get the word vaccine. That's pretty neat. So we can do vaccinations, so let's make an HIV vaccine. What's the problem with that? Well, it's kind of like the problem about these flu viruses. One of you said you got a flu shot recently, right? Do you have to pay for it? I don't know, I never got one. You have to pay for it? Okay, what if you do have to pay for it? Maybe it's expensive. I bet it's cheaper if you got the vaccine from 2013 because they got a lot of it around and nobody's using it. So it's probably cheaper to use old vaccines than the new one, right? Why wouldn't you do that? I'm always after cheap, good deals. Not wasting money on stuff. Why is last year's vaccine not as good as this year's vaccine? Huh? Yeah, so what people do is they try to make vaccines for the kind of flu virus that we're going to get this flu season. Okay, so they go to other parts of the world where these viruses are now, and they're causing problems, and so they try to figure out which one is going to be here in the future. And it's different from this flu virus we had the year before. Why? Well, the viruses change. Okay, so you really got to have up-to-date viruses. Well, that's because your immune system is fairly sophisticated, and it works by chemistry, and it recognizes the things that are the antigens that it builds antibodies to are surface proteins and surface molecules of these viruses. But those things are coded for in the genes, the genome of the virus. And so those things, as the genome changes, those surface molecules change, and so it's different recognition going on there, okay? But we can still make a vaccine for flu viruses and try to keep ahead of the game, but we realize that they're changing. The thing with HIV, that virus also changes, but I don't think anybody knows of another virus that changes as fast as HIV. It's changing so fast in an absolute amount of time that you couldn't come up with a virus to be ahead of that. So the virus, the vaccine thing looks like it's a wash and won't work for HIV. Let me see. Was it a big deal to figure this stuff out? You know, yeah, if somebody doesn't get the flu and you don't grow up, you know, at school someday. No, this is a big deal. There's a lot of people die in the past from diseases that we have vaccines for. Yeah. So up until I think 1972, people were still getting the smallpox vaccine, and that was a big disease that's virtually gone from the face of the earth because of vaccines. Okay, so this vaccination thing is a big deal and a major breakthrough. It just looks like it doesn't work for HIV. So what we have to do is just like fighting prokaryotic bacteria, you got to look for differences. Well, there's a big difference in HIV that we can exploit. Yeah, it uses our metabolic processes, it uses our energy, it uses our chemistry, it uses our molecules, but there is something that it does that we don't do. Did we say what kind of virus HIV is? It's a what? Yeah, it's called a retrovirus. Okay? So our genetic material is DNA, and we transcribe it, we copy it into pretty much the same language, and then we translate it into another language of amino acids, and those are the products that make me, and you, and me different than a housefly. Okay? HIV, its genetic material is RNA. And what it does is it takes RNA, and then after that goes into you, it also puts an enzyme into the host cells that's called reverse transcriptase. And it goes from RNA to DNA. Then that DNA goes into your genome, and then you go from DNA to RNA transcription, RNA to polypeptides, translation. We don't do this RNA to DNA thing, so there's something different that we can exploit, okay? So when you're building DNA from RNA, the building blocks are those nucleotides that come in 4 forms, and we use those letters of what? A's, G's, C's, and T's. Yeah. So what people did was they came up with essentially a molecule that's a fake T. Okay, so it's your processes that build DNA recognize it as being a T that should be used in building DNA from RNA, but when you use it, the process stops. And so if you put it in somebody that has HIV, they have the reverse transcriptase, there's the RNA molecule, but when they have these fake T's in there, it's something called AZT. Just think of it as fake T, it's called azithromycin. If you have that in there, the reverse transcription process stops, and it gets all messed up, and you're not able to replicate these HIV anymore. And what happens, if you were to take, make a chart, time down here and measure the viral load. It goes, let's say it's up here pretty high like this. You add AZT and the load of the viruses goes way down. You keep putting AZT, it works great. Okay? What happens over time? Oh, crap. Add more AZT, put more in there. You know, isn't that the way it works? If something kind of works at low dose, more is better, right? You can keep putting all this AZT in there you want, but the viral load goes up. What happened? Say, what did you say?
It mutated.
Yeah.
Resistant?
What?
They build resistance.
Yeah, what's that? That's a good word. What's this resistance going on?
The virus that isn't affected by AZT survives and reproduces and creates
Yeah.
more, so then it's not affected by it at all.
When this is an important thing. We put this in there. Did this make this virus mutate? No. It's always doing that. What does mutation mean? There are lots of things that we can think of as mutations. In their very simplest form, it's when, say, when you're replicating DNA, and if you want to use the word mistake, I don't know if that's the best word to use, but instead of putting a T, you put a C. It's different, okay? Does that make a difference? Well, sometimes no. Remember the redundancy in the genetic code, like 20 amino acids, but there's 64 codons. Sometimes you can change part of the codon, and it doesn't make any difference to the amino acid. Sometimes you change the amino acid that's there, but it's in some part of the polypeptide. It doesn't change its functionality. But sometimes, even the slightest change can make a change in an amino acid in a polypeptide, and now it behaves in a different way. Those are mutations. Just a base switch is the simplest one. But big pieces of DNA can come out, get inverted, get replicated. Any of these changes or inconsistencies in replicating genetic material we can think of as mutations, and they potentially result in not just genotypic differences, but phenotypic differences in these viruses. So they're always going on. Remember I said HIV is the fastest changing virus that we know? So if you think of some numbers here, in a human being that has HIV, they produce, as these viruses are reproducing, they make 10.3 times 10 to the 9 new viral particles every day. Equals what? Lots, okay? That this virus has a fixed mutation rate of about 3 times 10 to the negative 15 mutations per base per reproduction cycle, and its genome is only 10 to the 4 base pairs. If you look at those numbers, it works out that every day in a single infected person, there's at least one mutation at each nucleotide every single day in a person that's affected, okay? So, and it's not things like this that's making those mutations happen, they're always taking place. Now, can you do things to make mutation rates change? Yeah, there are mutagens, there are things that you can do to make mutations happen. They're not directed, you don't make certain kinds of mutations happen, but just go out and lay in the sun and do it every day, do it every week, and do it every year for a long time. If you have, particularly if you have fairly fair skin, what's going to happen to some of your DNA? Okay? Yeah, you're likely to cause mutation. Some of those mutations can result in differences that you don't want and result in things like skin cancer. Okay? So you can do things to increase mutation rates, but that's not what's going on here. Yes, mutations are always taking place, and the way that I like to look at it is these viruses are making tons of new viruses all the time. Okay? And these mutations are always taking place, so they're always making different kinds of viruses. Every generation, and they have very short generation times, you're always making a whole host of new kinds of viruses and dumping them out there into the environment. Every generation, okay? And they're all different. Some of them, okay, can deal with, they're not fooled by the fake T. So you start off with a bunch of viruses that are susceptible to AZT, but they're always making new viruses. Some of them aren't fooled by AZT, but there's no AZT around, and they're outcompeted by this old style one here. But you put AZT in there, and it knocks the population of the regular ones down, the AZT susceptible ones. But there are ones out there that can deal with the AZT, and they're not fooled by it. So who does better? They do. On average, the non-AZT susceptible viruses, on average, do better at gathering resources, existing, reproducing, and passing on the genes to new individuals that code for what? Not being fooled by AZT. So their population numbers do what? They go up, okay? And this is what you have here. This is a different virus than what we had over here. These were AZT susceptible, these are not. So how do we deal with that now? What's one thing you could do? You could stop the AZT, right? And often if you do that, the viral loads go back down, but then they go back up again. So what happened there? Well, you probably went back to this kind right here. They're still being produced, right? We're producing a variety of kinds of viruses. They still exist. And then in an AZT-free environment, they're out competing the non-AZT sensitive viruses, and so they're the ones that come back. How can we deal with this problem then? Well, you can give people this drug, and you can stop giving it to them. What's something else that you can do? Well, what people really do is, what if we gave them a fake G? Okay? So if you give them a fake G and a fake T, now you have to have the right differences in two places and not just one, and in two places at the same time. Are the chances of that greater or lesser than it happening in only one place? A lot less. Okay, so the way people deal with this is constantly change the environment. Okay, give them a fake one of these, give them a fake one of these, give them two fake ones of these, constantly changing the environment here. And so that people that have HIV, you can keep the viral loads low, but you have to keep constantly changing the rules of the game, and so they never get to build themselves back up. Okay? What's one of the problems with doing that? Costs money, you know? And so if you got a lot of money and you got HIV, you can kind of live with it. Most people that have that, though, they don't have money, and then they get HIV and they have it long enough that, remember, the target cells are the cells that run our immune system, and those things get destroyed, and then your immune system gets destroyed, and you die of fairly benign infections like fungi and bacteria and things like that, okay? Some important things here. Remember, this didn't cause those mutations. Those changes were always there. We just changed the rules of the game, and so different variants are better at dealing with a particular environment at a particular time, and it's those that do better relative to other things, and it's those that pass on the genes for those differences to the next generations. And then what we have is something different now than we had in the past. What did we call that the other day, last Wednesday? Transformation, right? It's one lineage of viruses going through time, and it's changing. It became something different. It went from AZT susceptible to AZT resistant. And because HIV changes so fast, okay, that building up that resistance, happens quickly. It's the exact same thing that takes place when you've heard of antibiotic resistance, right? So you can give somebody antibiotics to work against some bacterial disease, and sometimes they give you antibiotics, they say take it for 20 days. And you take it for 6 and you feel pretty good, you're going to say that for another time, so you quit, okay? That's not good. You're supposed to take it all the way through, okay? Also, just the widespread use of antibiotics. What you're doing is you're changing the rules of the game out there, and what's the term is you're selecting for antibiotic-resistant strains of bacteria, such that when you get one of those and you have to deal with it, that antibiotic won't work. So it's like selecting for an AZT susceptible a resistant strain, and then when somebody has that technique doesn't work, okay? So it doesn't matter, you keep pumping more on there, that even makes the problem worse. Then you're selecting for really tough strains of bacteria. The same thing happens with pesticides. So you have a field of corn, you get corn earworms in your corn, you don't like that, so you spray it with pesticides, and it knocks them down like crazy. Were all of those, these are caterpillars, the larvae of some moths, are they all the same? No, they're all different. Remember we talked about looking around the room, everybody in here is different. All those moths, they're different. Some of them can kind of deal with that pesticide. So on average, those do better at continuing to exist, gather resources, and reproduce more so than the ones that are susceptible to that pesticide. So over time, you can keep pumping more and more pesticide on that field, and you're just going to select for a very resistant corn earworm caterpillars that can deal with that pesticide. The pesticide didn't make the mutations happen. They were always taking place. There are always some individuals out there that could deal with it. If there were no individuals out there that could deal with it, they didn't have that much variation in them, what happens? Dead. Gone, okay? But in something like insects that reproduce quickly and they have huge numbers, there are variants out there that can deal with it. So you can build up AZT-resistant HIVs, you can generate antibiotic-resistant bacteria, and you can generate pesticide-resistant insects and other things. It's all the same thing, okay? It's transformation. It's the same corn earworm. It's still HIV. There's still the same kind of bacteria. It's just its characteristics have changed in that lineage over time. That's transformation. We're not dealing with diversification and multiplication of lineages. It's transformation of lineage. That's what's taking place. And we'll see that the mechanism by which it takes place is this thing called natural selection. That little scenario that I described to you of variant corn earworms and differential reproduction among those genetically coded variants results in differential reproduction and perpetuation of some individuals more so than others, and lineages, their characteristics change over time. That's how natural selection works. We'll spend a lot of time on this later on in the semester, but that's exactly what's taking place in all these situations, and that's what's going on here. Natural selection is taking place. This lineage has transformed, and we have something different now than we had in the past. Does that make sense? So this is an evolution problem. So once people realize that, now you know what you're dealing with, and you know you deal with ways that you can fight it. Another thing that we learned, remember we said you put this in here, now we get an AZT-resistant strain, we stop the AZT, and we go back and get our old kind back. Remember somebody over, you asked, was it you asking about something going back to something that you had before?
Yeah.
Yeah, that's just what we did there. And so one of the things that tells you is that this evolution, this process is not unidirectional. You're not going towards some goal, you're not always going in one direction. You can go back, okay? So you can either have those variants there, or if those variants aren't that different in the base sequence in their genetic material, you can get back mutations. So instead of putting a C in the place of a G by mistake, in the place of that C, you can put a G. You can go back to what you had before. That's a possibility. When you're making so many individuals, you can get back mutations to the old situation, okay? And so that's probably what's taking place here. So it's not a unidirectional thing. Okay, yeah?
Is there mutations that can cause problems but not always beneficial?
Well, in this case, it's all on the perspective. From the human perspective, that's not beneficial. If you were to sit down and talk with the viruses, that would be beneficial to them. Yeah, so it's all relative. So if we were talking about making value judgments on things being good and bad, we do those kinds of things with respect to people's welfare, but really, am I getting at your question?
No, I was thinking if there are mutations that are not beneficial to the organism, we might continue asking.
Oh, sure. And what, you know, if we have mutations in humans that result in not having any eyes, okay, that's probably a situation where that individuals that don't have eyes, with respect to our species and how it functions, you probably don't do as well as in the long term, on average, compared to other individuals that have eyes. And so, yeah, there are deleterious mutations, and natural selection tends to, remember, it's not an all or nothing thing, tends to work against those things. Am I getting with your question now? Okay, yeah. So that's a good point about mutations. It may be great to have some particular characteristic, but just because it might be good to have it, that doesn't make mutations happen that result in that. Mutations take place when later on we'll talk about how natural selection works. The mutation part is the random component of it. Okay? So these mutations are always taking place. That's what's generating these variants every generation. Okay? The natural selection part, look at the words. Is that random? No. It's anything but random. It's selection. Anytime you select something, it's based on something. That's what? That's not random. Okay? So we'll get to this again later on, but that's one of these misconceptions that I've talked about. People see something and you say, oh, that could never happen by random chance. You're right. You're absolutely right. It could not have happened by random chance. But natural selection is not a random process. The mutation part might be, but the rest of it is not. Okay? And that's how you can generate. That's how you generate complexity. That's how you generate adaptations. This AZT resistance, that's an adaptation. That's a characteristic coded for in their genes that give these a leg up on these things. They're better at that environment at this given time for that situation. That's what an adaptation is, and that's the only way we know for generating these things called adaptations, and that's natural selection. Okay? So, anything we need to clear up about what's going on here? We don't have to really solve our problem though yet, right? Lots of people still have HIV. If you have money, this is how people that have HIV for a long time deal with it. They just keep taking these different cocktails of drugs that keep changing the rules of the game, keep changing them fast. Okay, don't establish some rules of the game and let it play out for a long period of time, or there will be individual viruses that can deal with that, and their populations will build up, and you'll get problems. Okay. Remember we talked to the viruses. What did we ask them? Oh, if they, if this mutation or this change was bad for them. Some of them say, oh, it's good for us. Okay. So you've got this virus. It's in its host. It destroys its immune system, and it kills its host. Okay. If you could sit down with the virus and talk to it, what's another question you might ask it? Are you stupid? You're killing your hosts. This is your world. This is what you depend upon for your existence, and you're killing it. What's up with that? What does that tell? Does that tell you anything about the evolutionary processes? What were you going to say? Oh, that's a different part of the game. Yeah, spreading from host to host. But within that host, that's a dumb thing to do. Okay? But evolution can't see the future. It can't see what's down the road. Evolution works on what? The here and now. So these lineages are following what works good in the here and now. It may be stupid to kill your host in the long term, but evolution doesn't work that way. It works for the situation of the here and now. These populations are responding to characteristics of the environment at a given place at a given time. So evolution doesn't have foresight. You can't see into the future, can't predict the future. Even if you could sit down with paper and decide, oh, this would be a good way, wouldn't it be a good thing for it to keep the host alive longer? Okay? Sometimes that happens. That's a possibility that HIV, as it plays out in the history of humanity. One possibility is it wipes out Homo sapiens. Is that a possibility? Sure, I'm sure species have been wiped out, which is called extinction. Every member of that species is gone. That must have happened in the history of life on Earth by viruses. Okay? Another possibility is that as the viruses are producing variants, human beings are also what? Are we the same? No, we're always making different kinds of human beings. If there was a strong selective pressure of HIV, if there are variants of human beings that could deal with HIV, in the long term, what might you get? You might get Homo sapiens that can deal with HIV. They still harbor it and spread it around to each other. Okay, that's another possibility. That seems like that's what's happened with the simian immunodeficiency viruses in some non-human primates. They have the same kind of viruses, but they deal with it. Okay? Like what? No, they don't have. Well, they have. I think they have syphilis. Yeah, they have something. They have other things, but organisms often deal with these things. That's another possibility. You get. We'll talk about coevolutionary processes where two lineages are going through space and time together, and changes in one beget changes in the other, and changes in this beget changes in this one, and then they coexist. So that's another possibility of within the realm of possibilities. One of the points, though, is that evolution can't see into the future. Okay, it's also not perfect. Okay, again, looking at people, we have all kinds of problems that are products of our evolutionary history. But we don't have to be perfect, right? We just have to be what? Good enough. Yeah. You just have to be good enough than something else that might be competing with. And the same thing plays out here. These things aren't perfect. They can't see the future. And so killing the host may be a stupid thing in our minds, but that's what it does. Because it's successful, that strategy is successful in a different way. This natural selection is taking place in different realms. There's more than one game being played. So within a host, there is competition taking place among different kinds of viruses. Okay? So if you're a virus that's really strong and virulent, coming from the same word, very strong and virulent and reproduce very fast, and you outcompete other viruses within a given host, within that host, you will win. Okay? And the faster you reproduce, the more virulent, the stronger you are, you're also better at getting from host to host. That's the other game being played here. Competition within hosts, or being good within a host, but there's also the game of competition of spreading from host to host. Okay? Now consider two different kinds of people, not individuals. Remember, we're thinking what? Populations. So think different populations of people. If you have one, let's say we're talking about the spread of HIV through sexual contact. So if we have a population of people, and they tend to be very promiscuous and change their partners a lot, and you put two kinds of viruses in that population, a strain that's very virulent and a strain that's not so strong. It doesn't reproduce that fast, and it actually lets the host live for quite a long time. Okay? In a given body, which virus will win? The very virulent, fast reproducing, really strong strain. But it also kills its host very fast. Okay? In the situation where the host population is very promiscuous, is that a problem or not a problem? From the virus's perspective, it's not a problem. Who cares if I kill the host? I've already moved on to something else. Big deal if the host dies. Okay? Very successful strategy. So in a population of people that are very promiscuous and change their partners a lot, the virulent strain does better. okay? What if you only had the non-virulent strain? Yeah, it's gonna be the one that's gonna win, because it's the only one there. But head-to-head with a less virulent strain, the more virulent strain will win. What if you had a population of people that doesn't change their partners very much? They wait till one of them dies, and then maybe they wait a few years after that before they take up with somebody else. If you put both strains of that virus in that population of people, which virus, if you let it play out over time, remember, not within an individual. If you put both viruses in one person, the virulent strain is going to win, they're done. In a population of people, and you let it go through time, which virus is going to win there? The less virulent strain. Why? The very virulent strain that's in the population kills its host fast, but they never pass it on to anybody else. When they're dead, the virus is dead. They're ending, remember, they can't see the future. They're ending up wiping out their host before they have a chance to be passed on. The less virulent strain is tending to leave their hosts alive longer, and they're ending up in people that even one of their partners dies, they live long enough to take up with somebody else, and that virus ends up spreading. So we have two different games, we have two different situations here based on the behavior of the host population, okay? So what's one way we could, as human beings, deal with HIV? We could change behaviors. We could, as a population, select for less virulent strains. Does somebody's behavior change or make one virus become something else? No. Those different kinds of viruses are there. It's a population level phenomenon. You have to let it play out. Human beings are going to die. Okay? They're going to come in. Remember our lineage going through time. Individuals are dying, new ones are coming into the population, new ones are dying, new ones are coming in. But over time, the behavior of the population, of the host population, will select for different kinds of viruses. If you get that, you're getting some important things about how evolution works and how natural selection works. Okay? It sounds stupid to kill your host, but natural selection will select for that kind of viral behavior in a situation where who cares if the host dies? You've already moved on to 4 others already. Okay? So the viruses are competing in 2 levels, within a host and competing to get from host to host. Okay? And they're doing it at the same time. That's another thing to remember when we're talking about evolution, natural selection. They can't just play one game at a time. All these games are being played contiguously at the same time. There are many, many selective forces operating on these populations of viruses and these populations of hosts. And that's one reason you can't perfect dealing with any one of those problems, because you're dealing with all of them at the same time. Does that make sense? Okay, anybody got any questions on some of that? If you get that part about the different populations, you're getting some real important stuff for our class. Okay. Actually, there are some people that you can put the virus in them and they don't get an HIV infection and they don't get AIDS. Okay? And what's interesting is if you look at the world and you map the people that have this, they have this innate ability, this innate non-susceptibility to HIV. And actually, we figured out what it is. So remember we talked about cell recognition and your immune system and your antibodies recognizing antigens and things like that? Well, we're going to look at some pictures here. Probably we'll have time today, and we'll look at how viruses work. Well, they're very specific, and they recognize cells to attack by their surface molecule. Well, there are individual human beings because in their genome, in a spot, that there's a gene that codes for one of these cell receptors or co-receptors that HIV recognizes that says, oh, this is the kind of cell to attack. In that gene, they're missing 32 bases. And so it makes a different product, which has a different cell surface molecule, and when HIV comes up to it, it's the perfect cell to attack. Oh, no, that's not right. Oh, no, that's not right. Oh, that's not right. they don't recognize it as being the right kind of cell to attack. And so these people are immune to HIV. What a great discovery. Now you've got something that you can use against the virus. But an interesting thing is I'll show you a map, if you look at around the world where the incidence of this Delta 32 deletion that gives people resistance to HIV it's minuscule around the globe. But when you get up into Europe, particularly parts of northern and central Europe, I think it's something like 9% of the people have this base deletion, and they're not susceptible to HIV. Why one part of the world? Why that part of the world? Why would it? What might have given rise to that pattern? Why is it not the same all over the globe? Why is it not 9% all over the globe? Or why is it not minuscule to non-existent over the globe? Why is there a piece of the globe that has more than others? Got any ideas?
Yeah, I think it's like occurred in that area.
Yeah, but yes, but humans haven't had HIV for very long. I think there's a blood sample that people discovered in the 1990s. It's from 1959 from a man in Central Africa, and it's an old blood sample, and there's HIV in it. So there's something you can establish. In 1959, human beings had HIV. The other thing that we can do is we can go back and look at, hang on to your answer because you're on the right track, but it's not with respect to HIV. And I'm saying because it hasn't been around that long in people. You can also look at HIV and how much it's changed over time. And so what you can do is you can look at variability in modern day HIV, and you can look at its ancestry and look back in time, and you can figure out what's the rate of change in HIV. How long would it have taken for this much variability to have arisen by that rate of change? It's something that we'll talk about later in this semester called a molecular clock. So you can point back in time at when all these HIVs were the same. Of course, the farther you point back in time, your margin of error gets bigger and bigger and bigger. But there's a diagram I'll show you today that points back in time to the 1930s, plus or minus 10 years. So that's probably around when this virus jumped from non-human primates to humans. And since then, it's been spreading in humans and diversifying and gotten to the very diverse situation that we have today. So it really has, HIV is not the selective agent. What else took place, though, in big parts of Europe several hundred years ago? Yeah. Did many people die of that? Yeah, something 30, 40, 50% of the people died. Would you notice that? Yeah, I love it in my neighborhood. The streets are full of parked cars and the traffic and stuff coming to work. It'd be nice to, deal with 50% fewer people. But of course, some of those are going to be your friends and family, and 50% chance maybe it's you. Yeah, so okay, I'll deal with the parked cars and the traffic and stuff like that. But yeah, so the Black Plague, and there were multiple incidents of this parasite, this disease that's caused by Yersinia pestis, a bacterium, and it's one of the reservoirs are rats, and it's vectored by an insect, a flea, and moved from rats to people. And in parts of Europe where people lived in very close quarters, very close to rats, very close to each other, when that disease would break out, lots of people got it, and lots of people died. An interesting thing about the plague bacillus is that it uses the same receptors to recognize cells that it attacks. And so, yeah, one hypothesis, remember this is not a theory, this is a specific thing, one hypothesis for the distribution of that Delta 32 base deletion that gives rise to resistance of human beings to people from HIV is because it's the very same receptor that the plague bacillus was using to recognize cells in humans, and it tended to what? Wipe out more people that didn't have that base deletion than people that did. And so the people that did tended to, on average, live longer in the face of plague live longer, gather resources, and reproduce on average more than other individuals that didn't have that base sequence deletion. And so what happened to the frequency of that variant of that gene? It went up over time. And the other alleles relatively went down. So one hypothesis is that yes, it was natural selection that made the frequency of that base deletion happen in that part of the world under the pressure of the plague. So that's one possibility, okay? But once there are people that are not susceptible to that, that's something I think people are working on. And pretty soon, we can already do things like that. You can change people's genes, particularly if you're dealing with a zygote or a cell that the next generation would come from, and there's ways to edit genes. So why not change the gene in somebody that's at the one cell stage, and why not give them, make them not susceptible to HIV? That'd be a good thing or a bad thing? I don't know. That's not what science is not about, making those kind of decisions. Those are ethical things, and we draw those lines in different places at different times. Okay? Is that right? Is that an okay thing to do? I could, in 2016, I could probably argue it is. But we all, and you guys are gonna live longer than I am, and you're gonna draw the lines in different places, because what you're gonna see is all kinds of amazing things happen. So, an example I use is like, let's say I could grow extra kidneys in a jar, take cells from me and grow them around some matrix and grow extra kidneys such that if I lose it up for the next 50, I can make it 50 years, and I wreck my kidneys or my liver or something, I got a new one, and it's me, right? So I'm not gonna reject it, and I put it in and go about my business, okay? Is that okay with all of you? Is there anybody that doesn't like that? Well, it may be a possibility. Again, that's not a science problem, that's an ethics problem. I'm kind of okay with that. Yeah, I think that's kind of okay with me. Now, where I would draw the line, what if when I'm young, we grow two of me, or I grow a new one of me now, and the whole body is there, but you screw up the brain so it can't think and stuff like that. I draw the line before that. You see what I'm getting at? We draw lines in history in different places. Growing a new kidney, 100 years ago, they might have thought that was an abomination and horrific. I can live with that. Growing another one of me so I can take, like, when you buy an old car, it's good to get two, because then you can buy parts from it, to keep the one car running. It's the same thing. But me, in 2016, that's too creepy. I'm not going to do that. But there's probably people in here right now that would say, I got no problem with that, as long as you messed up his brain or something like that. So this editing of genes to make somebody so that you can grow people that aren't susceptible to HIV is well within the realms of possibilities. Well, you wouldn't know that without the perspective that we have from studying this problem. Okay? Let's see. Let's look at some pictures. If I can make this thing come on. Laptop. That. Translated and making new virus particles. It's pretty neat that people can figure out these things that you can't observe directly with your eyes. This is the same thing in a less complicated fashion, but you can see all the stages that are going on in the replication of HIV inside and to the detriment of one of these host cells. Okay? This is a good diagram. I hope you can see the different colors. We've got these are viruses. We can think of these are HIV. And we have a form that's here is just described as orange, okay? When orange HIV make new individuals, do they make new orange individuals? Sure, yeah, they do. So orange HIV gives rise to orange HIV. Always? No. Okay, remember those mutations are always taking place. Some of those mutations are such that they generate a different kind of HIV that's different enough that we'll call it this yellow form. Okay, or green. What do you want to call that? Green. Okay, so they're making orange and green ones. And as we go through time here, we're seeing that we went from one kind, we have two kinds here, and then those green ones beget green ones, but some of the green ones beget always green ones? No, they're always making differences. And here we got some gray ones. So we've got a situation where we have multiple kinds of viruses in a given host at a given time, and then we change the environment. We put in this AZT stuff. Okay? There's 3 different kinds of viruses in there. Are they all going to be able to deal with AZT in exactly the same way? No. Okay? Some are better at dealing with it than others. The ones that tend not to deal with it so well don't do very good at replicating, making more of themselves, and passing on the genes that code for those kind of viruses that don't do so well in the face of AZT. Other variants have genes that code for phenotypes that allow them to deal with AZT. On average, those tend to do better at existing, reproducing, and passing on the genes that code for the phenotypes for dealing with AZT. If you let that play out, what is this whole thing here is a lineage. We followed it through time. We started out with orange kinds. Over here we only have gray kinds. That's transformation. Okay? It happened, but it happened in the face of a selective force. We changed the rules of the game when we added AZT that's supposed to hurt these viruses, but some of them could deal with it. In fact, these gray kinds could deal with it. So even though at this time they were in the minority, after this situation, they are now in the majority. It's not. Is diversification taking place in this diagram? It kind of is, isn't it? So we've got transformation of this lineage from one thing to the other, but we kind of even did some diversification here from orange to orange and green. It's like, remember we said A can go to a fork and make A's and B's. It's kind of the same thing. It really isn't that big of a diversification that it's something completely different. It's still HIV, but we have made different kinds. But at a small scale, if we look at that lineage close, this diversification stuff is really going on all the time to generate all these variants that the environment selects for or selects against. Okay? That's where the term natural selection comes from. It's nature doing the selecting for among these different variants of things that are being generated through this variation generation process. So down here we see we started off with orange kind, and now we're in gray kind. We went from AZT susceptible to AZT resistance. Again, did the AZT make these gray ones come into existence? No. Okay, it just was a change in the rules of the game that selected for or favored that other kind. Okay, so there's always differential reproduction going on. Some things are reproducing at faster rates than others. These are the ones that reproduce at faster rates and pass on the genes for those characteristics that allow them to do that. And so these are the ones that become the prominent type in the environment at that time. Anybody got any questions on this diagram?
So what would be the right term for it? Would it be transformation or diversification?
Both. Yeah, it's really transformation because it's really one lineage, one kind of things. If this was people, these are all Homo sapiens. But really, if you wanted to, it's almost like we have 2 little different lineages in here. Okay, we'll see way later in the semester. What if we had different selective forces? Could we select for gray ones and orange ones with different situations? Sure, we could. And if we did that for a long enough time, what's gonna happen? They're gonna be different enough that we might call them different things, okay? In fact, that might be the next diagram. Oh. Read that all day. Oh, just so you have that diagram, here's absolute time. This is branch length. This is we're gonna talk about phylogenies. Remember these trees that show descent of lineages? Branch length, the longer the branches indicate more change from the beginning of the branch to the end of the branch. So this is this variability that we can measure in different pieces of time, and we make a best fit line here to point back in time to when all of these things would have come from, say, a common ancestor and been the same. And so this is where we get this estimate of human beings getting HIV in around 1930. But you can see the farther you go back away from your data, the margin of error gets bigger and bigger. So it's kind of like, oh yeah, it could have been as early as the 1920 or as late as 1940. But it gives you an idea that human beings have had HIV in them for less than 100 years, okay? Oh, we talked about where HIV, how we got it, and again, it's close association with human beings with other organisms. In this case, it's almost certainly killing and butchering other organisms that had similar kinds of viruses. So this is a bunch of meat that somebody's collected in Central Africa, and so there's a nice crocodile right here. We want to take that home for breakfast, and for dinner we could have gorilla. But I remember I said, you cut up these animals, there's two things. You're dealing with sharp knives, and you're dealing with blood. Okay? That gave a and then in the this century, so the 1900s, people have really been moving into parts of Central Africa and encountering and eating these animals on a much greater scale than they did in the past. And so it's given a much greater opportunity for this virus to jump from non-human primates into humans. And some of those variants, the greens, the grays, the oranges, could deal with people, and they became successful in people. And that's a good example of divergence, to the extent that we call one of them human immunodeficiency virus, and the other one, its ancestor kind of thing, simian immunodeficiency virus. Okay? This doesn't look too appealing to me. So, here's this diagram of the prevalence of that 32 base deletion that confers pretty much immunity to people that have it to HIV. And you can see around the world it's not very high, but this red color and red and yellows are higher incidence of it. You can see it's really centered up here, and this is also the place that was the most hit by those plague epidemics, and so that's one of the things, and that it's the same receptor that's used by the plague bacillus, which lends you to come up with that hypothesis that this change in the frequency of that allele here was driven by natural selection over the past several hundred years and gives rise to this interesting distribution of that difference among human beings. And then this is a very important diagram to illustrate multiple things. One is, remember in this class we think populations, and the other thing is we think trees, think bushes, okay? And so this is a phylogeny, or a phylogenetic tree, that describes lineages going through time. We could turn this way, and it would be like I drew it on the board, but it doesn't matter, we're drawing it this way. Time goes, oh, it's down here, it goes from the past this way. So what's taken place here? We have one lineage of things that diversifies and gives rise to two lineages. And then each of those gives rise to different lineages. What this is a phylogeny showing the evolutionary relationships of lineages of different kinds of viruses. Human immunodeficiency viruses and simian immunodeficiency viruses. This branching pattern is totally done based upon the base sequence of the genomes of those viruses. Okay? So, ones that have very similar genomes come together very quickly here. Because this is very similar to this. Okay? Things that join in very disparate places have very different genomes. So you can see in going through time, here is one kind of thing that diversified into two kinds of things, but it hasn't been apart that long. There, you would expect them to be very similar. It's that kind of information, again, we'll talk about this later this semester, that we use to build these trees. So a person built this tree based on the gene sequences in these viruses, so you get these relationships and these lineages for these viruses, and then on top of that, to learn something new, people superimpose the hosts. So we've got this virus here, and then you say, oh, what host is that kind of virus in? Oh, it's in the sun-tailed monkey, and so they wrote it down there. So we superimposed these hosts, so there are some monkeys here, there are some chimpanzees, and this kid with a bad haircut, in the case, human beings, and we, by doing this, we learned something really interesting and important. What can you gather from this diagram by the fact that humans are here, humans are here, humans are here, and if you look at that piece there, humans come out in a couple of different places there. What does that tell you? Has the virus jumped from non-human primates to humans only once? I think there's like 4 or 5 times, at least in this diagram here. That's the only way you can get this pattern. In this lineage up here of having humans over here, humans over here, humans over here. Okay? It's jumped multiple times from non-human primates to humans. If it's done it in the past, it can still keep doing it now. Okay? So that's one of the things that we learn from this. We'll also do this later on in the semester. We'll build phylogenies, and then these hosts have nothing to do with the shape of the tree. People built the tree first, and then stuck other information on top of it to learn something and look for patterns. And so these hosts were superimposed later to look at that and learn something about the history of these viruses. Okay? Anybody got any questions about this diagram? Again, it's the same thing we were drawing on the board the other day with time doing this. You can draw them on their sides. And again, these lineages are doing 2 things. They're transforming within a lineage, because this is different than this, and they're diversifying and giving rise to multiple lineages. And we draw them in these phylogenetic trees. Again, we'll talk about these and make them clearer later in the semester, but we use these things as tools to describe the evolutionary history of this group of organisms, or in this case, viruses. We'll stop there.